We described the common mechanisms of bacterial resistance to antibiotics. Now let's explain the mechanism of action and spectrum of activity of beta-lactam antibiotics. There are 4 classes of beta-lactams. There are penicillins, cephalosporins, carbopenums, and monobactams. And they are all bactericidal. What bactericidal means is that they actually kill the bacteria very fast. So as time goes forward, the number of colonies actually drop very quickly. As opposed to bacteriostatic agents that don't necessarily drop the number of colonies, but actually halt the growth of bacteria. So the number of colonies actually stay the same over time. Beta-lactams bind to penicillin-binding proteins, or PBP. And there are different types of penicillin-binding proteins which result in different spectrum of activity of beta-lactam antibiotics. Now, the first beta-lactams are penicillins, so these enzymes were named after the first beta-lactams, so they are called penicillin-binding protein, but they are also known as transpeptidase. And transpeptidase is an important part of cell wall synthesis. So by inhibiting penicillin-binding protein or transpeptidase, beta-lactams inhibit cell wall synthesis. Now, without going into too much detail, the synthesis of peptidoglycan involves several steps, and one crucial step is the cross-linking of components of peptidoglycan, and that cross-linking is done by transpeptidase enzymes, and that is the site of action of beta-lactams. Let's take a look at the spectrum of activity of penicillins. So here we are looking at, on the left hand side, gram-positive aerobes, so beta-hemolytic streptococci, Streptococcus pneumoniae, Enterococcus faecalis and Enterococcus faecium, as well as MSSA and MRSA. And then we have gram-negatives such as Haemophilus influenzae, as well as PEK, so P is for protease, E for E. coli, and K for Klebsiella. And then we have capes or space organisms, which refers to Citrobacter, Acinetobacter, Providentia, and Enterobacter. Then we have Pseudomonas aeruginosa. And then there are also major GI anaerobes, so Bacteroides is the number 1. And then we have atypical organisms. So none of the beta-lactams have activity against atypical organisms. Now let's take a look at Enterococcus. So for Enterococcus faecalis, you can see that basically penicillin G and V are different formulations of penicillin, and they are in general active against the wild type E. faecalis, but they don't have activity against E. faecium. Now the next 3 are basically have the same activity. So nafcillin, oxacillin, and dicloxacillin are basically in the same subclass. So when we test for susceptibility for oxacillin, it applies to nafcillin and dicloxacillin, and they do not have activity against Enterococcus. Then we have ampicillin and amoxicillin, which are basically the same activity, and they are just like penicillin, active against wild type E. faecalis, but they are not active against E. faecium. And then we have combination of ampicillin sulbactam as well as amoxicillin clavulanate, which has the same activity. And then we have piperacillin tazobactam, which again is active against wild type E. faecalis, but not E. faecium. And none of these penicillins are active against the VRE. Now for MSSA, because MSSA makes penicillinases, these penicillinases make break down penicillin, but they cannot break down nafcillin, oxacillin, or dicloxacillin. So nafcillin, oxacillin, dicloxacillin are actually considered drugs of choice for MSSA. Now those penicillinases can also break down ampicillin and amoxicillin. So ampicillin and amoxicillin alone are not active against MSSA. However, when you bundle ampicillin and amoxicillin with beta-lactamase inhibitors, then they have activity against MSSA because that penicillinase gets blocked. So the same applies to piperacillin tazobactam, this active against MSSA. None of these penicillins are active against MRSA. Now, gram-negatives for the most part make a lot of beta-lactamases, so you will see that majority of penicillins do not have good activity against gram-negatives until you get to the combination of beta-lactamase inhibitors. So that's when you start to get some activity against Haemophilus as well as PEC and Capes organisms. And the only thing that has activity against Pseudomonas in the penicillin class is Piperacillin-tazobactam. For Bacteroides, because Bacteroides also make beta-lactamases, only the beta-lactamase inhibitors are active against Bacteroides. Let's take a look at spectrum of activity of cephalosporins. Cephalosporins are broken into generations. So the first generation includes cefazolin and cephalexin. They are stable against the penicillinases that Staphylococcus aureus makes, so they are drug of choice for MSSA. So the higher you go to through the generations, the broader the spectrum of activity. So because first generations are the narrowest cephalosporins, you want to use the narrowest when it comes to MSSA. So cefazolin would be the drug of choice for MSSA infections. Now, ceftazidime in general is not good for gram-positive, so it is not active against MSSA. For pretty much all cephalosporins are not active against Enterococcus, with the exception of ceftaroline might have activity against E. faecalis only, but not E. faecium and not VRE. And the only cephalosporin active against MRSA is ceftaroline. And cefiderocol, which is pretty new to the market, is excellent for gram-negative organism, but absolutely no activity for gram-positive organisms. When it comes to gram-negatives, you should focus on what covers Pseudomonas. So ceftazidime, cefepime, and cefiderocol are the cephalosporins that cover Pseudomonas. And for Bacteroides, only the cephomycins, which are only a subgroup of second-generation cephalosporins, so cefoxitin and cefotetan, are active against anaerobes, Bacteroides specifically. The rest of them do not have activity against Bacteroides. And none of them are active against atypical bacteria. Now let's take a look at Carbopenum. So in general, we have ertapenem that does not have Pseudomonas activity, so it's narrower compared to the rest of Carbopenems. The rest of the Carbopenems are active against Pseudomonas. Now, all of them are active against Bacteroides, but none of them are active against atypical organisms. None of them are active against MRSA, but they are all active against MSSA. And lastly, imipenem might have some activity against Enterococcus faecalis. Recently, there have been a couple of carbapenems combined with beta-lactamase inhibitors. So we have meropenem verbactam as well as imipenem relibactam in order to maintain the activity of these carbapenem against carbapenemase producing Enterobacterialis, as well as multi-drug resistant Pseudomonas. Now, we also have a couple of cephalosporins bundled with beta-lactamase inhibitors. So we have ceftolozane tazobactam as well as ceftazidime avibactam. So ceftazidime avibactam is really targeted at organisms that produce carbapenemases, whereas ceftolozane tazobactam is directed at multi-drug resistant Pseudomonas. Now, one thing to note, these two are the only beta-lactam, beta-lactamase inhibitor combinations without activity against Bacteroides. So pretty much anything else that's combined with beta-lactamase inhibitor has activity against Bacteroides because Bacteroides make beta-lactamases. But ceftolozane and ceftazidime themselves, they do not have activity against Bacteroides. So Bacteroides has intrinsic resistance against ceftolozane and ceftazidime, so that beta-lactamase inhibitor does not remove that intrinsic resistance.